what was your role during the investigation, and what got you interested in being involved with the research?
Okay, so I was mainly involved in the microscopy side, so we were interested in where the site of action of this metformin drug, and we thought it might be in the mitochondria. So in order to prove that, obviously you then have to sort of look for localization of the mitochondria, and we used a reporter, a copper sensitive reporter, which showed the levels of mitochondrial copper.
How was the pi electron ring delocalization significant to the binding effects of metformin?
Well, as I understand it, I think that the interaction, the way in which metformin works is it's able to build a very strong affinity with copper, and is able then to pull copper off proteins, and it takes like 2 metformin molecules to form this ring structure, which actually is able to pull off the copper. So there's a very high affinity for copper.
So what was the experimental procedure for the separate experiment involving pioglitazone?
As I remember, I think the experimental procedures were pretty much the same as far as the microscopy was concerned. I mean, basically what you do is you take the cells and you load them with the copper reporter, and then you wash out the excess. It's a membrane permeable compound, so it loads into the cells, and it's a fluorescent molecule that becomes fluorescent when it loses fluorescence when it's in the presence of free copper, but once copper is lost or sequestered, then it becomes more fluorescent. So you load that into the cell, and then you administer the particular drug or compound that you're interested in, whether it's metformin or any of the other compounds that we tested. Some of them were simply positive controls that we knew would have an effect on copper. So we had to put some positive controls actually to prove that what the mechanism that metformin was working through. So essentially we had the compound, and then you take the cells, put them live in under a microscope, keeping them at 37 degrees and in 5% CO2 as they would be in the incubator, and then you could just image them fluorescently using fluorescence microscope.
What do these results tell us about metformin's effect on type 2 diabetes, and are there any side effects?
Well, what it tells us, I think, is that metformin is not working through the mechanisms that were originally thought, or at least it's not solely working through those mechanisms. So what it gives us is an opportunity to try to develop new compounds which might be based on metformin, which may, for instance, have a better copper sequestering ability than perhaps metformin. So it's now we understand at least slightly better the mechanism by which metformin works. It allows us then to take metformin as a lead compound and design other drugs around that compound to get a better, more efficient drugs. Maybe, as you say, drugs with a lower, less side effects, or the side effects of metformin are not so serious.
So what results from the experiments can be further used in order to help with the treatment of type 2 diabetes?
Well, essentially, that's what we'd hope to do. I mean, we'd hope to demonstrate that we can understand fully the mechanism. I mean, we don't actually have, we haven't got every nut and bolt in place in terms of the mechanism of action of metformin. It's only a preliminary publication. So we hope we'll get more idea of what effect it has on the mitochondria of the cells, whether it actually affects their dynamics or their positioning or their fission and movement in the cells, and then. We then have a sort of bioassay in which we can test other compounds. So we can design other compounds, or chemists can design other compounds based around metformin, which may have a much better activity, and we can test those in a sort of an in vitro bioassay on cells, which then obviously gives us some clues to how much better they work.
Okay. And how can this study be used to further understand the effects of other drugs and their metal binding properties?
Well, I mean, these fluorescence indicators. There are other indicators around for other metals, and so you could potentially look at other compounds using exactly the same type of assay. So you could actually use sensors for, say, zinc or for lead or any particular metal that you're interested in, and then you could then test other drugs and see whether they were working through the similar mechanism of action. And
for other diseases as well, like cardiovascular diseases and
yeah, I mean, if that's where the way that some of the other drugs are working, I mean, there are drugs related to metformin which might be working, maybe working through exactly the same mechanism, which maybe not through sequestering copper, but maybe sequestering other elements which are necessary necessary to the cell. Okay.